Hi guys, we are the Kraken, and our prompt statement ID is SIH25042, and our prompt statement is identifying taxonomy and assessing biodiversity from eDNA datasets under the Ministry of Earth Science. Our goal is to revolutionize deep sea biodiversity analysis. We leverage cutting-edge AI to identify taxonomy and assess biodiversity from eDNA, unlocking the secrets faster and more accurately than ever before. Why is this revolution needed? Because the current methods used for marine discovery are held back by these three critical points. Deep sea inaccessibility. Exploring these habitats is difficult and expensive, leaving a reference database of known species dangerously incomplete. Or missing unknown. Current analysis pipelines are designed to find what they already know. They often discard DNA sequences from the unknown species and effectively throwing away potential discoveries. And the database bottleneck. The reliance on reference databases makes the entire process incredibly slow, which is very inefficient, and which creates an obstacle for large scale biodiversity monitoring. Here is our AI based pipeline, which solves the pain points effectively. So, to begin with, we receive the eDNA sequences from the lab in the form of FASTQ files. Then, after that, we apply methods like trimming, sequence filtering, and techniques like ASV and DDA2 are applied. After which, we'll take the DNA sequences and pass it through the DNA BERT model. The DNA BERT model will convert the DNA sequences into meaningful embeddings. Then we'll pass the embeddings to our XGBoost model, which would give us a result with its confidence. If the confidence is higher, then we'll classify it. If the confidence is lower, we'll pass it through unsupervised learning pipeline, which will later give us an file cluster, which will be our candidate taxa. Then, to avoid false positive, we'll verify the candidate taxa with the BLAST database. And if the candidate taxa matches to the BLAST database, then we have got a red flag. So we'll send a feedback loop to our XGBoost model through a reinforcement learning. If it doesn't match, then we found novel taxa. So after which we'll estimate its abundance, diversity indices like Shannon's, Simpson, richness, and also calculate scientific outputs like species matrices, diversity matrices, biodiversity hotspot, and risk assessment. Now coming to feasibility analysis. Proven models and a clear advantage. Model we used have been tested on real-world dataset, showing a strong performance in taxonomy assessment. The software we are using, including the models, are open source, which completely eliminates the licensing costs. Compared to traditional methods, you can see in the radar diagram that our AI approach is significantly more time efficient, accurate, and excelling at novel species discovery by reducing the dependencies completely. And it also decreases the dependency on database as compared to the traditional pipeline. This allows us to see what others miss, providing a more complete picture of marine diversity. Now let's answer a few frequently asked questions. Why did we choose advanced models like DNA-BERT2 and DLUCS instead of relying on conventional architectures such as CNNs? This is because DNA-BERT2 gives us state-of-the-art accuracy in DNA understanding, and DLUCS enables unsupervised clustering to discover new species. Unlike traditional architectures like CNNs, which struggle with genomic data, these models are far more effective for our goals. The second question is, since advanced AI models require high performance GPUs, how will our solution remain accessible to labs or institutions with limited computational resource? We have built solution for that. We offer lightweight CPU based fallback pipeline, which includes models like DNA Grind and XGBoost that ensures any lab can use our system efficiently and effectively, even without an high-end GPU. Coming to the third question, how do we prevent AI from making errors? Like, firstly, identifying a novel species that doesn't actually exist. So, for this, we are using a two-step safeguard method where, uh, after getting the candidate taxa from the unsupervised learning pipeline, we'll cross-verify it from the BLAST database. And if it matches, then we'll send a feedback loop to the XGBoost model through reinforcement learning. And if it doesn't match, then we have got a novel taxa. This is the way we handle false positives. Now coming to the fourth question, we have highlighted the potential of for a single microbe discovery to create a multi-billion dollar industry. How realistic is this? And is a project success dependent on such a rare find? No, in short. Breakthrough discoveries are possible, but not our core dependency. The real value in our platform lies in consistent outputs like reliable biodiversity mapping, sustainable fisheries management, and climate monitoring. A rare microbe discovery would be high impact bonus, but the solution is already impactful through its long term and practical benefits. Now coming to the fifth question. Given that software components are open source, what are the primary costs associated with running our platform? Uh, our primary costs are for processing power and storage, but since we are avoiding licensing fees, the platform remains highly cost effective and very scalable. Coming to the sixth question, how exactly does this project contribute to India's blue economy? So, to begin with this, right now, India doesn't have its own independent deep sea biodiversity database. Our solution helps fill the gap by generating novel taxa records, species matrices, indices directly from the eDNA sequences. These outputs give researchers and policymakers reliable data for sustainable fishery management, which is a critical since fisheries already contribute about 1% to India's GDP and are valued over 20 billion USD. By enabling science backed conservation and fishery planning, our project directly strengthens India's blue economy. Here are a few more FAQs. You can please go through it. Thank you.